The plasma membrane. The plasma membrane, if you recall from the last slides, is the outside layer, the outside sort of squidgy barrier between the inside of the cell and the outside world. In eukaryotes, in animal cells, it is composed of a lipid bilayer. So there's one layer and there's two layers, and it's made mostly of lipids. The lipid bilayer is made up of molecules that have a hydrophilic head, represented by these pink balls, and hydrophobic tails, the squidgy blue guys down there. Most of those lipids are phospholipids. So you have a phosphate-based head, which is hydrophilic, and a carbon chain, a saturated carbon chain, coming away from the exterior of the cell. Remember, out here is aqueous. It's water-based. It's a polar environment. The inside, oops, sorry, these are probably not showing up very well for you, is also water-based. They're aqueous. So when you have two layers, each of which has a hydrophilic head and a hydrophobic tail, in an aqueous environment, the head is going to be attracted, being philic and having polar molecules, is going to be attracted and capable of forming even brief interactions with the water. But the phobic stuff doesn't really have anything in common with water. It only has something in common with other phobic stuff. So when you put a molecule that's amphiphatic in water, that means it has a philic head and a phobic tail, or a philic side, a hydrophilic side and a hydrophobic side. They will spontaneously arrange themselves so that the phobic stuff touches phobic stuff and the philic stuff touches philic stuff. I do tend to abbreviate hydrophobic and hydrophilic as phobic and philic a lot in these lectures. It's a bad habit that I picked up from an old professor of mine. After this unit, you're free to refer to it as phobic and philic purely, but for the time being, for the purposes of the first exam at least, I'll need you to write out hydrophobic and hydrophilic, okay? So some of these are phospholipids, some of these are glycolipids. Those are going to look similar to the phospholipids, but they're going to have a sugar group as a head, and that might even stick out a little bit onto the outside of the cell. Those sugar groups have branches, and they can become entangled with one another if they're dense. This forms something called, this sort of tangle of these sugars that are sticking off of your glycolipids forms the glycocalyx. The particular molecular arrangement of which is something that your immune system can recognize. It's unique to you, and it's unique to each one of your tissues, the cells of each of your tissues. So your immune system can come along, take a look at one of those cells, read what's on the glycocalyx and say, oh, cool, that's Rhiannon's red blood cell. No harm, no foul. I'm going to keep moving on. If it finds something that has a different glycocalyx, then it knows that it's foreign. It doesn't belong there, and it's going to try to attack it and get rid of it somehow. I say somehow because different immune components act different ways. Now, I drew those on very, very tightly packed, but in reality, only about 5% of your lipids in your lipid bilayer are these. So your branching is going to really kind of be broad and not terribly dense. You'll also find cholesterol in the lipid membrane. Okay, these little yellow tags here. We'll talk about their importance in a moment in a little more detail, but these are embedded throughout the phospholipid bilayer in the phobic portion because cholesterol is hydrophobic. It's a hydrophobic molecule in and of itself. It helps anchor the phospholipid together So it is the two layers of the bilayer are attracted to one another spontaneously like this. But it helps to have some staples in there, right, to help keep it together a little extra much. The cholesterol helps do that. And it also helps provide a little bit of extra rigidity to the outside of the cell so that it's not completely shearable with any incoming force. Okay, so here's the lipid bilayer again. I'm just going to stick with this one image for these sets of slides so that we can have a little consistency. But you have the lipid bilayer again. Here's the philic head, phobic tail, philic heads, phobic tails. You have cholesterol, these yellow guys that are going to help anchor the lipid bilayer together. You can see it pretty well on this one right here. How you have these phobic rings. Those are carbon rings. So they're just carbons with a bunch of hydrogens on them. That's super phobic, right? No oxygens, no nitrogens, no nothing to be found. There's that. And then you have this like zigzaggy tail drawn on it. That's another carbon chain that's also quite phobic that would ordinarily branch like into the other side and help keep it together. In addition to those, you will have other protein complexes that have other purposes aside from just forming and maintaining a barrier. Protein complexes that are embedded into the lipid bilayer that cross both layers, like you can see this guy does, is called a transmembrane protein. And since it is embedded in that plasma membrane, it's called an integral protein. These integral proteins, whether they're transmembrane or not, some of them are not, have hydrophilic and hydrophobic regions to keep them aligned properly. So if you have a protein that's embedded right here, and you examined it chemically, the part of the protein that's up here is going to have a lot of hydrophilic stuff going on so that it can intermingle with the it has to be hydrophilic so that it can intermingle with the hydrophilic heads of the cell membrane. And because it's going to be touching the hydrophilic water-based environment outside the cell. This stuff, I'm going to use a different color because why not? Down here is going to end up being pretty hydrophobic if you examined it chemically. If it were hydrophilic, the force of these guys not having anything to do with it and the pull of the philic stuff would pop this protein right out without a lot of anchoring. If it were entirely hydrophobic, the aqueous environment of the cell and the outside hydrophilic part of that membrane won't have anything to do with it, and it will get pushed in the middle. So to be embedded properly and touch any of those surfaces, you need a hydrophilic and a hydrophobic end. And that arrangement of which side is philic and which side is phobic is going to help keep it oriented properly in the membrane. Integral proteins are permanently anchored into the cell membrane, so they're not transient, they're not going to come and go. And they can serve a variety of functions. They can be receptors, so they can be, you have a lot of sugar tags drawn on here, but you might have, like, let's pretend that guy's a receptor, a little nub of something that's going to be capable of receiving something from the outside world. That receptor doesn't change. It stays in the same place on that same cell as long as that cell is alive. It's permanent. It's a permanent integral protein. It could be used for attachment. So some cells have to stick on to certain surfaces or other types of tissues or other types of cells. They all have to touch something for the most part. So it can be an attachment protein. You don't want an attachment protein to be temporary, right? If you're going to anchor yourself somewhere, you want to stick around. Those are going to be permanent structures as well. They're typically attachment molecules are typically integral proteins. They could do some enzymatic activity. So we're going to find later on that we have some proteins, and you might remember this a little bit from A&P, that you have membranes or you have proteins built in to that plasma membrane that sort of, when they're activated, they can sort of spin a turbine and make ATP. That's integral. That's integrated into the plasma membrane, into the lipid bilayer. It's got a functional part that sticks out into the cell, and it does, and it has enzymatic activity. It facilitates the adding on of a phosphate group to ADP, adenosine diphosphate, so that it can go forth into the cell as ATP, and then that can get split somewhere else to do work. They could be involved in transportation, so import or export of stuff. The pore here that's drawn is a good example of an integral protein. If you see a pore here, it might be something that lets things come and go fairly freely as long as they're a certain size. It might be selective and only let certain things come in, or it might be selective and only let certain things come out. And they may be involved in cellular communication. If this cell needs to talk to a cell nearby, maybe there are two neurons that has to transmit its signal from one neuron to the next. Maybe this cell is super sick and needs to tell the ones around it, batten down the hatches, I'm about to die, and whatever's killing me is going to get released into your neighborhood. Cells can sometimes sort of send these signals to one another. The proteins can help facilitate that transfer of information. There's a couple different things that a couple more things that you find on the plasma membrane. One category is called cell adhesion molecules. These are sticky glycoproteins found on the surface of cells that have some sort of anchor to them. We're not talking about free-floating cells so much, we're talking about cells that have a place in a fairly otherwise solid-seeming tissue. They can help the cell anchor itself to the extracellular matrix, so basically keep its place within a tissue rather than just sort of being floating and bouncing around amongst a bunch of other floating, bouncing around cells. Sometimes they can help migrate themselves across other cells. For example, you remember proteins can denature, they can change shape. Sometimes that's permanent, sometimes it's temporary. If this cell, let's say it has a cell adhesion molecule, interacts with one from another cell, that's terribly drawn. If the interaction between the two causes this to change shape, you may be able to propel it along in a certain distance across another cell. It can also be a way of cells sort of sensing things going on in the environment. Manipulation of the cell adhesion molecule lets the cell know that there's something going on its surface. This is a really common and oftentimes promising target for cancer research. So cancer cells are really just terribly mutated, terribly meaning just in a bad way, not a lot. Mutated cells that you already have in you. It's in a way your body turning against itself. When the cell mutates to behave a certain type of cancerous way, for it to spread and for it to metastasize usually involves changes in these cell adhesion molecules, so that this cancer cell's relationship to the cells around it are different than the relationship of regular cells to other regular cells. By having a drug that targets those cell adhesion molecules, you could potentially kill off, or not kill, but get rid of those molecules or inactivate those molecules. So maybe you don't kill the cancer, but you don't let it go anywhere. Or it could provide a way of anchoring onto the cell adhesion molecules in such a way that the cancer cells let the drug in, which then kills the cancer. So that's a really popular target for cancer research. Finally, we have proteins on the cell surface, just in general, that are part of its relationship to the immune system. This is separate from the glycocalyx, or rather it's in addition to the glycocalyx. These proteins are interspersed along the surface. They're not unique phospholipids with a sugar head like the glycocalyx was. These are separate integral proteins that are embedded into the plasma membrane. And their function is to hold out on the surface infectious agents for immune cells to recognize. So the glycocalyx is part of the cell's signal that, hey, I belong here, don't kill me, to the immune system. Major histocompatibility complex isn't directly recognized by the immune system necessarily, But what it'll do is it'll hold out foreign things like bacteria or viruses or whatever, proteins that it finds in the environment that don't belong there. And it holds it out, waiting for an immune cell to come pick it up. When that immune cell comes by and recognizes that that head on a stick is sticking out on top of the cell, it's going to swipe up that foreign thing and then do with it whatever that immune cell is going to do with it. Again, there's a bunch of different kinds of immune cells. They all do different stuff. So we'll just keep this broad for now. But it also has to recognize that that MHC protein, that major histocompatibility complex protein, isn't part of the problem. So it does sort of recognize it, but that's not really the point of it. It's more to distinguish it from bad stuff rather than to create a self-identity. Now, this is something that we need to match for transplantation purposes. The glycocalyx is a really complex set of sugars on the cell surface or that are part of the lipid bilayer. The major histocompatibility complex is integral to the plasma membrane, but not necessarily part of it. It's really easily, well, not easily exactly, but it is much more readily characterizable than anything having to do with your glycocalyx, and it's an awful lot more unique than any part of your glycocalyx. So if you're transplanting a tissue into a person, you're going to match up a lot of different factors in their body. You're going to want the blood type to match. You're going to want, I don't know, you need a lot of things to match. One of those things are the proteins within the major histocompatibility complex. If the proteins on the incoming tissues of the MHC are different enough from your own self, your immune cells aren't going to know that those new incoming proteins aren't part of the problem, and it'll attack. The closer you have a match between the MHC protein the MHC proteins on the incoming cell surface to yours, the better tolerated that tissue would be. This is why whenever anybody needs a transplant, the first place they look is the person's genetic family, the person's biological family. They're most likely to have the closest possible match to not only your blood type, or at least it's quite likely that they'd have the same blood type as you, but all those other proteins are way more likely to match up with yours than anybody else on the planet. So the biological family always gets looked at first. And then if nobody is able or willing or whatever to donate their tissues, and sometimes they can't look at the family because, like, what are you going to take your living dad's heart? He needs that. So then they go out into the community and organ donor cadavers and try to match those up as closely as possible. Finally, we're going to talk quickly about junctions. These are how cells can be stuck to one another. There's three major categories of tight junctions that we need to learn about: tight junctions, desmosomes, and gap junctions. Tight junctions are integral proteins of one cell that fuse with integral proteins of the other cell. They merge, they covalently bond, they are stuck together like glue. This is an extremely, extremely strong type of junction. This junction that's created is impermeable, and it encircles the entire cell. They're arranged in these lines. Oh, I'm going to get rid of the blue because that matches a little too closely. Let's go with this orange. In these lines across the cell surface, like quilting. If you go, into your bedroom and take a look at a quilt, you can see how the stitches on one side are the same on both sides. It goes through both layers completely and then circles back up. It interweaves through. But in this case, it's more like if those stitches were little tiny staples that keeps them together. This space in here between those two plasma membranes that are stuck together by tight junctions is impermeable to the rest of the environment. Stuff isn't going to get in between those two. In addition, stuff isn't going to be able to get through between those two plasma membranes unless there is a special lined up set of integral proteins otherwise that specifically allow communication. Desmosomes are anchoring molecules. They firmly keep one cell next to each other, but it's not rock solid, it's not super tight, and it allows for some flexibility. So what you have is this big, like button-like plaque right here on the inside, the cell interior side of the plasma membrane. They have some sort of proteiny tails that float to the interior of the surface, and they have other molecules, other proteins, that emerge to the exterior of the cell. Those get locked together sort of like Velcro. So they're stuck together right in here somewhere, basically. But just like if you take the two sides of your shoelaces and you lace them, or the two sides of your shoe that's laced together, and you lace it fairly loosely. If you hold each side, you can shift the fabric up and down, in and out a little bit, but you can't really separate them. In a similar way, those proteins on the exterior side of the cell that entangle with the proteins from the desmosome on the other cell allows them to be attached to one another in such a way that tension's distributed fairly evenly. No one part of this is getting pulled on more than another part. And they can move a little bit. You can allow a little bit of movement without tearing or shearing, basically breaking this part of the plasma membrane. If you were to apply the same sort of force here and here, this tight junction area, the plasma membrane, might just rip. If you apply that same type of pressure to an area that's locked together with desmosomes, or between two cells that are almost exclusively connected with desmosomes, it won't rip. You'll have some shifting. Things will bounce back into place. Nothing will tear. Finally, gap junctions are made of rings of tight junctions that have a little hole in the middle. That hole that gets formed, that pore, allows molecules to travel between one cell to the other in a really efficient, seamless way. They may be somewhat selective for certain molecules, but if you're the right kind of molecule, you can move from one cell to the next pretty easily based on diffusion or voltage gradients. So over here, you can see this a little bit, how there's an opening on one side, I'm sorry, I know it's small, and an opening on the other side of that gap junction. I could just go whoop, right through. It's literally like a door or a window between the two cells that lets things through. Let's think of a couple of different tissues and how these might be employed. These types of junctions might be employed. If you have a skin cell that absolutely needs to be attached to its neighboring skin cell firmly and completely, which one of these junctions would you want? Or let's take like two kidney cells or something that doesn't move. What type of junction would be best? Right, you would want tight junctions. You want one cell to stay locked down in exactly the right orientation next to the next cell. There's no movement that you have to worry about resisting. There's nothing else going on. You just need to stay locked tight next to something else. You're going to go with tight junctions. What about something that needs to move a little bit? Anything with movement is going to want to have some desmosomes. And a lot of tissues towards the exterior of your body, tissues around joints and stuff, are going to have a lot of desmosomes. What about tissues that need to transmit, say, electrical signals or chemical signals between cells very efficiently? Yeah, you would use gap junctions. It has a hole in the middle that's made to move molecules. Now, no cell only has one of these types. You really have a mixture of these types throughout. The ratio of which types you have can be pretty different, but you have, at least two of these represented on most cells. So let's think about a combination here. Your heart. What does your heart have to do? Well, what's cool about the heart is it has its own intrinsic rhythm. You don't need your brain to be working for your heart to be pumping. Your heart can pump itself. It's really cool. If you get heart stem cells and you grow them out on a dish, like in a heart cell specific petri dish, once you grow enough of them, they'll just start beating on their own in the dish. It's really cool. Your heart pumps itself. So what it has to do is it has to generate a signal, transmit that quickly to all the other cells of the heart in a direction. Like, it can't just go randomly around. It has to go in a direction and be coordinated. And that has to cause movement of the heart. Would the heart cells fare well if they had lots of tight junctions? No. First of all, tight junctions, the gap junctions are made up of a bunch of tight junctions, but just having tight junctions there that are impermeable doesn't help anything travel. Also, if you need movement to happen, you really run the risk of just tearing. What about desmosomes? Desmosomes keep cells together, but allow for movement. So does that sound like something that a heart might want? Yeah, a heart pumps. A heart physically moves all the freaking time. You need to be able to allow movement and of cells sort of past each other a little bit. You don't want to pull them out of alignment, but you do need a little bit of give so that you don't tear anything on that cell. What about gap junctions? Do you think it'll have gap junctions? Yes. Your heart, remember, had to make a signal and then send that signal. That signal has to be of something, electricity or molecules or whatever. In order to distribute that signal quickly, you want fairly low obstruction to that process. So why not just open up some doors? Why not just make little portals for molecules to move between one cell to the next? If you were to examine heart tissue very closely, what you would find is these sort of, and I'm going to draw them very linearly. They don't really look like this, but as a model. You would have these cells, and I'm not going to draw the nucleus or anything like that, next to each other. You would find desmosomes abundantly here in between each sort of layer of cells. You would find lots of gap junctions between the cells so that the signal can run along their length quickly and efficiently. And as the heart moves, this cell and this cell has desmosomes to make it so they can shift a little bit. But they still stay next to each other, and they can always, but they can still shift a little bit.